Aging is the single greatest risk factor for late-onset neurodegenerative diseases, particularly for a class of disorders known as tauopathies. Now, studying a disease process that can take decades to develop in humans presents a substantial challenge for researchers. So how do we investigate this? Well, a key strategy is to model the aging process itself in experimental animals. In this analysis, we're going to dive into the scientific review, Can accelerated aging models inform us on age-related tauopathies, to understand how both accelerated and even decelerated aging models are being used in the search for the molecular origins of pathogenic tau. So, here's our roadmap. We'll begin by establishing the fundamental link between the aging process and the development of these tauopathies. Then, we'll explore the different models of accelerated aging that scientists use to try and mimic this process. After that, we'll look at the other side of the coin, the insights that can be gained from organisms that exhibit decelerated aging. And finally, we'll conclude by examining the current limitations of these models and discussing the future outlook for this critical area of research.
All right, let's start with the core problem. The single biggest challenge in understanding many of these neurodegenerative diseases is that their prevalence is directly and powerfully correlated with chronological age. This isn't just a coincidence. It is the central observation that requires explanation.
And you can see this relationship very clearly in the epidemiological data. This chart reveals a sharp, positive correlation between age and disease prevalence. Whether we're looking at a primary tauopathy like progressive supranuclear palsy, where tau is the main pathological protein, or a secondary tauopathy, like Alzheimer's disease, where it's a key feature alongside other pathologies, the trend is undeniable. As the population ages, the incidence of these diseases rises.
So what exactly is a tauopathy? It's a neurodegenerative disease defined by the intracellular accumulation of insoluble aggregates of a protein called tau. Under normal physiological conditions, tau is a microtubule-associated protein. Its job is to stabilize the cytoskeleton, which acts as the transport network inside our neurons. In a disease state, however, tau detaches from these microtubules and clumps together into what are called neurofibrillary tangles. This process leads to synaptic dysfunction and eventually neuronal death.
Tauopathies are broadly classified into two types. On one hand, you have familial tauopathies. These are caused by direct mutations in the tau gene itself, which is known as MAPT. These are hereditary and they typically manifest at an earlier age. In contrast, and really central to our discussion, are the sporadic tauopathies. These are not caused by a single known gene mutation, they appear later in life, and their causes are much more complex and multifactorial. And it's this complexity that explains why age itself is considered the most significant known risk factor for this much more common form of the disease.
This brings us to the models. Since studying normal aging in common laboratory animals is such a time and resource-intensive process, researchers need faster methods to recapitulate an aged cellular environment. This is precisely the rationale for using models of accelerated aging.
A primary strategy here involves the use of what are called progeroid syndromes. These are rare human genetic disorders that cause phenotypes of premature aging. By introducing the causal genetic mutations into animal models, scientists can create an aged cellular environment in a fraction of a typical lifespan. This provides a system to study how these age-related cellular changes might contribute to disease.
Now, these progeroid models generally fall into two main categories, as shown in this table. The first are laminopathy models, which involve defects in the nuclear lamina. That's the protein meshwork providing structural support to the nucleus. The second category consists of DNA repair deficiency models, where the cell's innate ability to fix genetic damage is compromised. But I want you to pay close attention to the key limitation column here. For both categories, a direct, spontaneous link to the kind of tau pathology we see in human sporadic disease is consistently missing. Let's dig into why that is.
Let's start with those laminopathy models. A classic example is Hutchinson-Gilford progeria syndrome, which is caused by a mutant protein called progerin. Now, what's interesting is that a specific microRNA known as MIR9 naturally suppresses progerin expression in neurons. This means neurons are endogenously protected from this particular aging-related protein. Even when scientists artificially force neurons to express it, they observe nuclear distortion. But, and this is the key part, no apparent tau pathology or cognitive decline. This is a crucial finding, as it suggests that simply disrupting the nuclear structure is not a sufficient trigger for tauopathy.
And we see a similar story with the DNA repair deficiency models. In mouse models of Werner syndrome, for example, cognitive and motor deficits are present, but tauopathy hasn't been reported. In certain models of xeroderma pigmentosum, there is clear neurodegeneration, yet spontaneous tau aggregation is not observed. In Cockayne syndrome models, While there are indicators of neuroinflammation and axonal damage, a direct causal link to tau aggregation in a living animal remains elusive. The recurring theme here is that while these models do recapitulate aspects of aging and neurodegeneration, they don't seem to recreate the specific conditions that trigger sporadic tau misfolding.
It's not just about progeroid syndromes, though. Researchers also use other valuable models. Clotho-deficient mice show cognitive impairment, which reinforces the hypothesis that this gene is neuroprotective. The SAMP8 mice are particularly interesting because they are a polygenic strain, meaning their rapid aging is caused by a combination of multiple genes. This may better mimic the complex genetics of natural aging. And while they do develop some features like phosphorylated tau, they don't form the mature neurofibrillary tangles seen in human disease. Then you have the African turquoise killifish, a naturally short-lived vertebrate that provides a powerful system for studying how protein aggregation changes across an entire, albeit very compressed, lifespan.
So if accelerating aging doesn't fully recapitulate the necessary conditions for tauopathy, what can be learned from the opposite approach? Let's now consider organisms that exhibit decelerated or even negligible senescence to see if we can identify potential protective mechanisms.
And this question brings us to a compelling alternative model system. The star of this research is the naked mole rat. As the longest-lived rodent known, it provides an incredible natural experiment in resistance to the common diseases of aging.
The naked mole rat displays a suite of exceptional traits. It can live over 30 years, which is about 10 times longer than predicted for a rodent of its size. It's highly tolerant to physiological stressors like hypoxia, it's remarkably resistant to cancer, and, most pertinent to our topic, it shows a very low susceptibility to the kind of protein aggregation that defines diseases like Alzheimer's and other tauopathies.
Now, here is the most striking finding. The naked mole rat's tau protein is 95% identical to human tau. They have abundant levels of it in their brains, and it even undergoes phosphorylation, a chemical modification often considered a prerequisite for pathology. And yet, it remains correctly localized in the axon and does not form pathological aggregates. This strongly suggests the naked mole rat has evolved unique, protective cellular mechanisms, perhaps superior proteostasis or chaperone systems that prevent its tau from misfolding. Identifying these mechanisms could provide critical insights for therapeutic development.
All right, let's synthesize these findings. We've examined models that age rapidly due to specific genetic defects, and we've looked at a model that ages exceptionally slowly due to natural adaptations. We now need to assess what this all means for our ability to understand sporadic tauopathies.
This brings us back to the central question of the review. Given all this evidence, can these accelerated aging models truly inform us on the molecular origins of sporadic tau disease, the form that affects millions of people around the world?
The crucial takeaway is this: there are major limitations to our current models. As this slide summarizes, none of the standard models we've discussed spontaneously develop the hallmark neurofibrillary tangles that define human tauopathies. The accelerated aging they produce, which is typically driven by a single powerful mutation, just doesn't fully capture the complex, multifactorial nature of true physiological brain aging. The cellular environment created by these models is simply not yet sufficient to trigger that initial critical misfolding event that starts the disease cascade.
And that quote really encapsulates the current challenge. While these models have taught us a great deal about the cellular consequences of aging, they have not yet revealed the precise origin of the disease itself. The future of this research will lie in developing new, more sophisticated models that can better replicate the subtle, complex, and interconnected changes that occur in the human brain over a lifetime. The ultimate question remains, what is the precise combination of age-related factors that finally pushes the tau protein over that pathological edge? Finding that answer is the key to unlocking the mystery of sporadic tauopathies and developing truly effective treatments.